Cool. Yo, what's up?
Hey, what's up? What's up? How you doing?
I'm good. I got my internet,
Yo.
got my desk.
What's up?
Hey, what's up? What's up?
So I'm
Good.
back in business.
Up and running. Up and running. Did you get the stuff I emailed you?
Let me check.
This one shouldn't take anything long. At least the genetics PowerPoints that we just had to go over aren't very long.
Lab things. Oh, electrochemistry.
Yeah. Shouldn't be that hard, I don't think, but I would just have problems with half reactions and stuff still.
Mm-hmm.
And I've got a date for finishing chemistry 2, so finish it by August 15th.
Got it.
So, and then for the test Wednesday, you'll come to the house?
Yeah.
Okay.
Yeah,
I'm
back
I
in,
sent you the thing. You read it, right?
Yeah. Wait, what's up?
I sent you the thing on messages?
Yep.
The email?
Got it.
Yeah. So,
Yeah.
6 short answers, 10 multiple choice. Hour and 40 minutes. It starts at 4, not 5.
Okay. All right. So basically, I could share something with you where I go over these electrochemical cells, like the theory behind it.
Okay. Oh, let me give you access to this. There you go.
Here, I'm gonna share something with you. You see this?
Hmm.
Do you see the screen I'm sharing?
Yeah.
Okay, so this is just like some theory behind it. So basically, this is how batteries work, right?
Okay.
So essentially, you have you're basically. Let me zoom out for a sec. So in a galvanic cell, aka an electrochemical cell, aka a battery, we take advantage of the natural difference in the reduction potentials of two different metals to spontaneously, which means you don't have to add energy, it does it without needing energy, to spontaneously generate an electric current. So we have here, a zinc electrode, all right, and it's connected with a current carrying wire to a copper electrode,
Hmm.
and so basically the electrons travel from something that has a low reduction potential to a high reduction potential. So the half reaction here for zinc has a reduction potential of negative 0.76, whereas that for copper has a reduction potential of 0.34. So since the reduction potential for copper is greater, right?
Yeah.
That is the one that gets reduced, aka gains electrons. So the zinc has a lower reduction potential, so it loses electrons, aka gets oxidized, and electrons are moving here. See these light green arrow over here? That's the transport of electrons, aka electricity.
Okay.
So electrochemistry, measuring the concentration of copper within a concentration cell. So that's what they're essentially doing. They're using basically the same thing, except instead of zinc and copper, they're using copper and silver.
Okay.
So they're basically saying that, so they're basically, it's the same kind of thing over here. Piece of copper, right, in a copper sulfate solution, right? And oxidation occurs in the copper half cell. Oxidation, remember Leo and Gur.
Yeah.
So oxidation means loss of electrons, right?
Yeah.
Good.
Reduction's the gain.
Correct. So the electrons are going to be transported from the copper electrode to the silver electrode. Okay? And the anode, think of anox and red cat. So wherever oxidation happens, we call that the anode. So this copper is called the anode. And, wherever reduction happens, red cat is the cathode. So the silver electrode is the cathode, okay?
Okay.
Now, so at first there's a positive sign on the cathode and a negative sign on the anode.
Okay.
But since the anode, right, is losing electrons, right? That anode starts to become positive And the cathode, since it's receiving electrons, starts to become negative.
Okay.
So when that happens, right? So okay, as that happens, they complete the circuit with that salt bridge. So the salt bridge allows the nitrate ions from that silver nitrate solution to travel to the, to the other electrode, and it also allows the copper cathode. Copper cations to cross the salt bridge and enter the opposite electrode. So
Okay.
that's just a way to keep the current flowing.
Got it. Okay.
Yeah. That completes the circuit, allowing a constant flow of moved charged particles, aka, electricity, right?
Mm-hmm.
Now, okay, so basically, then they talk about volts and things like that. Let's see. Voltmeter, blah blah blah. Things about hydrogen. Yeah, overall difference in the overall you know um like electric potential of the cell is basically the addition of the reduction potential and the oxidation potential. So that's the overall,
Okay.
yeah, that's the overall cell potential.
Got it.
So, also, the reduction potential, or is just the negative version of the oxidation potential, and vice versa.
Okay.
So, okay, so for the half reactions that they give us, right? They're all reduction potentials because they're giving us. I mean, it's gaining an electron, right?
Yeah.
Now we use the one with a higher reduction potential. So, when we have silver and copper, which one has a higher reduction potential?
Mm, um, Silver and copper. Shouldn't it be the copper most of the time?
Well,
Or
look at
or
page 3.
the what?
Oh, page 3.
Share the screen again.
Oh, I'm no longer using that. I'm using the thing that you sent me, the electrochemistry PDF.
Okay.
Yeah. So
Oh, okay.
on page 3 of that,
Okay.
you see like half
Oh
reactions.
yeah, the standard reduction potentials. So
Yeah.
okay.
So which one is higher?
Oh no, it's gonna be the well, wait, if we're talking about reduction, it's supposed to be negative, no?
Oh no. Just these are already reduction, so don't worry about that yet.
Oh, okay, so then it's a silver.
Silver, right? So silver has a higher reduction potential, right?
Uh-huh.
That means that it is the one that gets reduced, and the thing with a lower
Hmm.
reduction potential gets oxidized.
Okay.
So the copper, the reduction potential is positive 0.34. That gets
Uh-huh.
oxidized, so we're going to say that that oxidation potential is negative 0.34.
Oh, okay.
And then we draw it the opposite. We draw it in reverse. Like copper solid turns into copper 2 plus ion plus the 2 electrons.
Okay.
Yeah. So the balanced chemical reaction, if you add the two, it looks like what they have down there.
Okay.
You see the 2Ag plus the copper solid, yields 2Ag solid plus 2, copper 2 cation.
Okay.
All right. So
Got it.
yeah. So then the delta G, that's the Gibbs free energy. Remember that?
Mm-hmm.
So this is basically a formula to basically find the cell potential under non-standard conditions, because standard conditions means that uh, there's just one molar like concentration of all of this stuff.
Yeah.
So, you know, so we use this equation, and E is a cell potential, F is Faraday constant, doesn't change. N is the moles of electrons transferred in the balance equation. So, in this case, it's whatever, 2.
Mm-hmm.
Um, okay, so so if everything's at standard conditions, we use that circle symbol that looks like a degree symbol.
Yeah.
All right. And then non-standard conditions, we use basically delta G is equal to the delta G under standard conditions plus RT ln of Q.
Okay.
Yeah. And, you know, then you get this thing, it's like whatever. It's called like the Nernst equation. You get basically the cell potential is equal to the standard um, state cell potential minus RT over NF ln of Q, and Q is just the concentration of the copper ions to the Ag ions.
Okay.
Yeah. Um. Okay. So basically, um. Okay. Then it talks about just redox equations. Reactions happening. Um. And this is you plotting the cell potential versus the natural log of the lower potential of the or rather the half cell of the copper when it's a lower
Mm-hmm.
potential.
Okay.
Okay, then let's see procedure. Um, Copper sulfate and magnesium sulfate. Use a serial dilution. Okay. Dilution equation, which you should know by now. Okay. And then place tube 1, which is, the least dilute, next to tube 6, which is the most dilute, in the front row of your test tube rack. Place a length of clean copper wire into each. The wire should protrude from the tube by several centimeters. Don't allow the wires to touch each other. Make a salt bridge from a flexible plastic straw and a length of twine, which is just, some type of string, They use that for, like, cakes and stuff.
Mm-hmm.
Pull the straw out and bend the straw over into the U shape and cut the... Alright, so just telling you how to make the salt bridge, essentially. You fill the tube with the magnesium sulfate. Make sure there are no air bubbles in the liquid. Quickly turn the salt bridge so that the open ends are facing down. Okay, blah blah blah. Right? Okay.
Mm-hmm.
Use the voltage device. So that's just going to be something that it's going to be in the data sheet that they sent us.
Mm-hmm.
And it's going to be a potential source of error. So but anyway. Yeah, the voltage displayed should be a very small number. And then you should say 0, so you're tearing it, essentially. TARE, right?
Mm-hmm.
So, okay. So basically, like, like, since since we're testing basically if the salt bridge is in contact with the liquid on both tubes. So that's where we
Mm-hmm.
record the voltage of the first cell. And then we basically, do that with test tube 6, and then we can replace 6 with 5. So then it can be 1 and 5, and then we, measure the new cell potential. Should it be higher or lower, right? So we're going to do that then with 4, test tube 3, test tube 2, right? And we're going to see, the difference in potential, voltage essentially. Then, after that, we basically, make a new cell using an unknown copper concentration. And using that, we find, the cell potential.
Okay. Got it. All right. So I was doing the Excel sheet over here. Film it out. Okay. Good. You there?
Mm-hmm.
Oh, okay. All right. What are we up to over here? I'm just putting some experimental stuff down. This is from the data. I think.
Yeah, you got number 20.
21.
21, okay.
21's my number, yeah.
And these are values for the voltage?
I believe so. Should be.
Okay.
Yeah. So how are you finding these concentrations?
I just opened up a spreadsheet and did the math that way.
Oh, okay.
Like, for some reason, I don't know why Google Docs doesn't let you use the spreadsheet.
Okay.
I'm trying to put these like side by side.
Hmm. Well, let's see. Um,
Whatever.
Why did nothing allowed to go message her? So you're completely moved out of the other place now?
Yeah.
That's good. So you only live in this place by yourself, right?
Mm-hmm.
Ah, okay. Will you have to roommate with anyone?
No.
Oh, okay. That's pretty cool.
Mm-hmm.
So I finished my history course with a 99.
Congratulations. That's really good.
Yeah. In this class, I should be getting an A in.
Mm-hmm.
This chemistry lab class. Genetics, if we're able to ace that test, possibly with a bell curve, I can get an A minus or something. Or I don't know, in the science realm, they probably just give A's.
Mm-hmm.
It would just be an A or something if it's over 90. But if anything, B plus can definitely happen. Or B. So, but I think the fact that their short answers might be good
Mm-hmm.
allows for explanation. So I'm guessing since it's not letting it do, just expand it. Do this.
Alright, I'm gonna have to do some of this shit in Excel. One sec.
Oh, okay. I had. Do you want me to do it, or like, or you got it?
Oh, what?
The stuff in Excel, because I copy and pasted the table here. I wasn't sure what to just do yet.
Here, one sec.
what you're up to on the Excel?
Yeah, I'm trying to make this graph over here.
Ah, okay. Okay.
Do they give you like a spreadsheet to help with the making the line of best fit?
No, they didn't.
Okay.
They just told you to prepare a graph here with in the PDF at the end of calculations. That's how they tell you to get the line of best fit.
Okay.
Equation given by Excel is in the form of y equals mx equals b. What's going on with the stuff there? Is this what we're supposed to actually be doing?
Well, let me see something. You could see here. I'll share it with you, or rather, I'm trying to see why I'm not getting consistent numbers. ln of this. Let's see. This to this to this to that. ln of C. Concentrations just keep decreasing. That makes sense. Let me see. Okay, 1 sec. I see. Times C2. Alright, now I finally got what I was looking for. Now I just have to edit this a bit. So let's see. E3. Okay. It gets more the lower. Okay. Lower. C2. Okay. Gets more and more negative. That's weird. With all right, I'm almost done.
Okay.
Trend line, okay. Horizontal axis. Okay. Vertical axis. Okay. Bold it. Bold it. Got it. Okay. Okay, I'm almost done. Okay, let me now just copy or save this. Okay. Do you see that?
Yeah, I do. Perfect.
So that's the graph of cell potential versus the natural log of the lower concentration of copper.
Okay.
All right. Now, using this, we're going to find the concentration of copper in our unknown solution.
Okay.
Let me do like the superscript stuff. For some reason, it doesn't let me do it.
Alright, so you get this first step to the introduction.
All right, whatever. Okay, so now we do it for the unknown and then see where it fits.
Okay. Okay, so let's see the short introduction. Okay.
Okay, I think I know what to do now. Okay, I think I got it.
Okay.
Um, it's yeah, it's the trend. It's a trend line. So I got the here. Let me copy this instead. Okay. So the so I put in the values for everything, and that trend line, that line of best fit, the equation for that is above in, y is equal to mx plus b form. So now we know what the m is, we know what the b is, and I just have to plug in the y, which is the e cell, the cell voltage, to find the copper concentration.
Got it. Okay.
Yeah. So okay. So from here, I'm gonna copy this equation over here. Okay. So okay. Okay. Let's see. Okay. Oh shit. Wait. Got it. Oops. Damn. Three, okay. Line of that. Okay, alright, awesome. Okay, now one more step. Okay. Got it, okay. Use a measured value of that for the unknown and the values of that from the equation of the line. Okay. So measured value, okay. Right. Measured value. So E3. Okay. Okay, E3. Awesome. okay, wait. Let's see. Got it. Okay, never mind. Okay, I'm almost there. Give me a sec. Mm-hmm.
No problem.
Okay. Okay.
Since we started at 2:30, can we do till 6 today?
What's up?
Or can we do till 6 today?
Like 2 more hours? Okay.
Well, because we started at 2:30,
Mm-hmm.
around there almost, like, so it's already 4:30.
Mm-hmm.
I just wonder if we can get an extra 30 minutes.
Sure.
Yeah, okay. Because there's gonna be 30 minutes less on Wednesday, most likely. So.
Got it.
This test is only like an hour and 40, something like that. So it'll make up in like 3 hours in those sessions, those 6 hours in total. It's a little more complicated than I thought. Well, it was complicated. How do you make those symbols? Like the E?
Insert special characters.
Oh, okay.
me use the bathroom real quick. Okay, I'm back.
Okay. Finish the introduction here. So what's next?
Well, what'd you say?
I finished the introduction.
Okay. All right, the discussion part. Let me open up this lab formatting thing again. Oh, whoops.
Oh, how to write a lab report? Did you save it? I'm going to send it.
Wait. Okay, yeah, it's in my email. Let me just look you up.
Oh, okay.
I got it.
Perfect.
Okay, discussions.
Discussion is basically a rundown of what happened, including any sort of experimental error. Yes, let me see what I can do. So the concentrations of the Cu2 plus and the unknowns over here, which are the
Yeah.
results, are this, right? What I
Yeah.
highlighted? Okay. Okay.
Okay.
Yeah. All right. And I did the abstract, so it works.
Okay.
So all done.
All right.
Can you download it as a PDF? Download the doc too. Alright. Let's see. Now, I'm gonna start sharing my screen.
Okay.
And yep, that's finished. Okay. Gene regulation eukaryotes. So, okay. Is it?
Yeah.
Okay. Is that good size?
Yep.
Okay.
All right. We have some fruit flies there. Okay. All right. Enhancers. Got it. All right. Chromatin structure in eukaryotes often makes the DNA unavailable to transcription machinery. Additional RNA. Okay. Processing events. Okay. Transcription and translation are separate. They're compartmentalized. And gene regulation controls cellular differentiation. So, all right, we can move on to the next. All right, so these are, okay.
Wait, sorry.
Yeah. So this is, just an overview again. So DNA, right, gets transcribed into primary RNA, right?
Yeah.
And then to become mature mRNA, right? What's done to it? Like, what are those?
Sorry? Say one more time.
Yeah, in the primary RNA transcript, what are those purple segments?
The purple thingies?
Yeah.
Aren't those introns?
Correct. Good. So the green things are?
Exons.
Good. So that's the splicing, the alternative splicing.
Yeah, the alternative splicing.
Polyadenylation is what happens to that 3 end. So that's where you have all the A's.
Mm-hmm.
And then the 5 cap is on the 5 end, and it just, it's basically to protect it.
Okay.
Then it, leaves the nucleus. And, RNA has a short lifespan, so it can, a lot of it will degrade. But the ones that don't get translated, right, into some type of protein, right?
Yeah.
And then there's going to be modifications, post-translational modifications. So that can, include either some type of thing added on one side, that blue thing, or something added on the other side, the yellow thing, or it can be degraded, or, it can be localized in a specific region, or it can, be active like an enzyme.
Okay.
All right. All right, so these are the different RNA polymerases. So polymerase 1 it transcribes the ribosomal RNA genes, so basically the ones that make up the ribosome. 2 transcribes the genes that encode proteins, right? 3 transcribes the tRNA genes and some small RNAs. We're going to focus on polymerase 2, which is the one, that basically transcribes genes that encode the proteins. Okay, so promoters. What's up?
No, I'm saying promoters.
Mm-hmm. Yeah. They're
we talked about them in TATAs.
Yeah, the Tata box.
Yeah.
Yeah, so they're going to be adjacent to the gene. And they bind the RNA polymerase. And, they allow like a basal level of transcription, just like a regular amount, I suppose, of transcription without it being, enhanced or inhibited or something like that. So, you have like different, examples of that down there.
Okay.
Yeah. The enhancers, all right, they could be very far away from the gene, and they either augment or repress the basal, which is the regular level of transcription. So they could be either at the 5' end or the 3' end of the transcription site, Like 5' end means it's upstream, it's ahead of it. 3' end means it's downstream, it's, behind it. If it moves to different positions, it's still going to function. Okay. All right. Now, we could identify enhancers by using reporter genes. So, for instance, you can construct a recombinant DNA molecule that has a putative enhancer sequence fused to a reporter gene such as GFP, which is green fluorescent protein.
Yeah.
So let's say that we have. Okay, so let's say that we have a gene that is transcribed at high levels only in the adult eye. So you have that adult eye specific enhancer up there in the purple.
Yeah.
And this causes transcripts only in the adult eye, right? Now we can do this. We can throw in the GFP sequence, so we could throw that in front of the promoter, and it'll start to make a basal level of, which is a normal level of GFP transcripts in all cells, and it can make that GFP protein undetectable. So basically, it's going to always be there, but we're not going to be able to see it basically because it's always there. It's in all cells, right? Now, the reason that we use these reporter genes is like the thing on the bottom right, where let's say we have that adult eye specific enhancer, we place a GFP gene there, and then it, instead of having a basal level of GFP transcripts, it has a higher level of GFP transcripts, and not in all cells, but in just the cells of the adult eye. So instead of it just making, a single protein, it's going to make several GFP proteins.
Okay.
So that is how and that's going to be, fluorescing green. So we're going to be able to tell where the location is of the, adult eye specific enhancer, like that gene.
Okay.
Okay.
Ooh.
So yeah, here's an example, right? There you go. Okay, now insulators, right? They're sequences located between an enhancer and a promoter. they block access to the promoter. So first we have, an example of an RFP gene, and
Okay, so red
yeah,
fluorescent
there's a
protein.
promoter in that direction, right?
Mm-hmm.
And then, there's a GFP gene and promoter in the opposite direction, and the enhancer, they basically upregulate the transcription of these genes.
Okay.
But an insulator causes the opposite. They block access to the promoter. So the enhancer, when you see the
Mm-hmm.
thing underneath, if you place an insulator between the enhancer and the RFP gene, the enhancer is blocked from accessing the promoter of that RFP gene. So in that case, only the GFP gene is transcribed.
Okay. Okay.
All right, insulators organize genomic DNA into loops. So, they bind CTCF proteins to form loops called topologically associating domains, TADs. And enhancers activate promoters located on the same loop. So we have an enhancer, that thing, whatever, the circle with the A there. And
Mm-hmm.
it's attached to that promoter 1. So it's going to activate that, right? Just because it's in
Yeah.
that loop. However, since it's not in the other loop, since promoter 2 is in the other loop, right? It's going to not activate that.
I see. Okay.
Whereas if you got rid of the CTCF proteins, right, they would all be part of that same loop, and promoter 1 and 2 would be activated.
Okay.
So it's just a way to create, to separate the loops. Okay. Now, so transcription factors, they're basically just a protein that binds to a specific, DNA sequence, right? So they can bind to promoters, they can bind to enhancers, and they essentially just start the transcription process going by recruiting other proteins to influence transcription, like, RNA polymerase 2 and stuff like that. There are 3 types: the
Okay.
basal factors, which is just normal, like, basal metabolic rate is like your normal, resting, metabolic rate, so just that's normal. Activators cause it to be above normal. Repressor causes it to be below normal.
Okay.
Yeah. So transcription factors are identified using GFP reporters. So mutations in a gene encoding an activator reduces the expression of the reporter. Now, mutations in the gene encoding a repressor increases the expression of the reporter. So basically, if there's a mutation in something, it's going to cause it to work in the opposite way. Right?
Okay.
Like, so like, all right, we have a wild type where there's that yellow circle thing. It's basically, something for, an activator, right? It's a gene activator. And,
Yeah.
it causes, a lot of GFP to be produced. Now, if there's
Yeah.
a mutation for that activator, it's not going to do that. It's going to basically reduce the expression back to normal, or back to basal levels.
Okay.
Okay, basal factors. So, but they bind to promoters of protein encoding genes. Ordered pathway of assembly at promoter. So the TBP, I think that's like the whatever TATA binding protein or something. It binds to the TATA
Mm-hmm.
box. So, you see it basically, up there.
Mm-hmm.
Yeah, it binds to the TATA box. the TAFs, they bind to the TBP, and then RNA polymerase 2 binds to the TAFs. So you see that it's like a couple of proteins that are involved here. So, okay, a mediator a mediator is a complex of more than 20 proteins. They don't bind to the DNA directly. It bridges the RNA polymerase 2 at the promoter and activator or repressor proteins at the enhancer. So there's an example of basically the entire complex. So basically what happens is the mediator, right? So the TBP binds to the DNA, right? The TAFs bind to the TBP, and the RNA polymerase 2 binds to the TAF. And the mediator basically binds to the RNA polymerase 2, and it can bind to either an activator, in this case it's an activator, or it can bind to a repressor at the level, at the DNA site, that sequence that is the enhancer. So it's basically all of that stuff that can cause the transcription.
Okay.
So activators are responsible for much of the variation in the levels of transcription of different genes. So, on the left we have basically the same example as before, where it stimulates recruitment of basal factors and RNA polymerase 2 to promoters, because it's an activator. And what it does, like on a DNA like molecular level, is that it recruits coactivators which open the chromatin structure, making it more exposed, right? Remember that about chromatin? Like, the beads on a string, the nucleosomes, right?
Yeah.
Like, more condensed means that less of it is exposed and less of it is transcribed. But when they're less condensed, right, and open, it's going to be transcribed more.
All right.
Okay, domains within activators. So activator proteins have at least two functional domains. So there's the DNA binding domain, which binds to the specific enhancer. The activation domain, which binds to other proteins. They could be coactivators, which cause the chromatin to decondense, right, to spread out more, or basal factors, which cause it to kind of be repressed and go back to normal. They can also contain a dimerization domain, which allows them to create a dimer, like to interact, to cause a connection with another protein.
Yeah.
So these are some examples of activators. Examples of the DNA binding domains. So there's the helix turn helix and the zinc finger. So these fit within the major groove of the DNA. So you see basically. So first let's look at the helix turn helix.
Mm-hmm.
So we basically have those cylinders essentially that that bind to it, and so the helix turn helix and the zinc finger are just binding domains for the activators to attach to. Okay. Yeah. Okay. Dimerization domains. So, the homodimers are, when they're made out of the same subunits. Heterodimers when they're made out of non-identical subunits. So, we have, a homodimer, which is made of something that starts with
the same thing.
Yeah. And these are examples of leucine zippers.
Okay.
Yeah. Okay. Repressor proteins. Okay. So just like an activator can recruit co-activators, right? Repressor proteins can activate or can recruit co-repressors. So that's going to do.
Wait, which one do the binding ones get?
Oh, the activators.
I mean, the activating ones.
Yeah, they do the reverse. So the activators recruit co-activators.
Co-activators. Okay.
Yeah.
Yes.
These do the opposite. So they're either going to prevent, directly prevent the RNA polymerase II complex from binding to the promoter, or they'll cause the chromatin to condense. Yeah.
Okay, so they'll cause them to condense, but then the activators will allow them to spread out and open up. And
yeah, the
co-activators will what do you call it? Let the RNA2 complex bind to the promoter.
Yeah.
Okay. All right, so just the opposite.
Mm-hmm. The same transcription factor can play different roles in different cells. So let's say we have, what is this? Okay, so we have the same transcription factor, which is the blue thing, dorsal protein, right? Now, in gene 1, it can act as an activator. So it has a co-activator attached. It recruits a co-activator, and it makes, transcription of that gene go up. Whereas it can act as a repressor for gene 2, right? Which means that it connects to a co-repressor and it stops the transcription of that gene. So, yeah. Now, repressor proteins can act through competition with an activator protein. So, an indirect repressor interferes with the function of an activator. So, this can happen due to competition due to overlapping binding sites. So, we essentially have, on that enhancer, right, we have the activator and the orange thing or whatever is the indirect repressor, right?
Mm-hmm.
So that's where they compete for that same site. So the indirect repressor, the orange one, got there first before the activator, right? So that's that's competition. If they bind to one another, it's called quenching because they cancel they cancel each other out.
Yeah.
Now, it can also bind to the activator and keep it inside of the cytoplasm, therefore, preventing it from entering the nucleus. So that activator, right? It can the indirect repressor, the orange thing, can bind to it in the cytoplasm and keep it from entering the nucleus and causing whatever protein to be transcribed.
Okay.
So that's cytoplasmic sequestration, and then the last thing it can do is it can bind to the activator and prevent it from dimerization.
Okay.
Yeah. Okay, histone modifications affect transcription. So we kind of talked about this before. So histone acetyltransferases, right? That they
Mm-hmm.
acetylate the histone tails, right? Which basically opens up the chromatin, causes more gene expression. Um, and many co-activators are these histone acetyltransferases. Um, the opposite are histone methyltransferases, which cause methylation and make the uh, chromatin more closed up. And so, these can activate or repress transcription. So some of them are co-activators, some of them are co-repressors. Um, yeah, and they're dynamic. They can be modified. Like the modification that they do um, can be very fast, very quick, reversible, etc.
Okay.
Okay, complex regulatory regions enable fine tuning of gene expression. So, in humans, about 2,000 genes encode transcriptional regulatory proteins. Each regulatory protein can act on many genes. Each enhancer has binding sites with varying affinities for activators and repressors. So, okay, we have basically an enhancer. So, okay, in the eye, in an eye cell, right? Whatever, in the G1 phase, um, you have you basically have, let's see, phosphate hormone. Um, okay, okay. So okay, you basically have um, I guess you have like, let's see. Okay, so G1 phase, whatever, it doesn't matter because nothing, there's no like transcription or whatever happening in the G1 phase, right? But in the S
Hold on.
phase, yeah, in the S phase, we have.
Yeah. Or a tiny bit later. So,
Oh yeah, how are we on
well,
time? Okay, so yeah, basically whatever. This is just saying that, um, you know, sorry. So we have like transcription in the eye during the S phase, and whatever because it's phosphorylated. But a hormone can cause in the skin enhancer, which in the skin cell, basically, it has a greater affinity for activation. So that's basically what it's just saying.
okay.
Okay. Now, so the function of a transcription factor, like whatever, changes by allosteric interactions. With, for example, a steroid hormone receptor binds to an enhancer only when bound to the steroid hormone. Also by modification of transcription factors like phosphorylation, and also transcription factor cascades. So first we have an example of the steroid hormone. So the receptor, without the steroid hormone, the allosteric effect isn't like, see that, like the shape of that green thing.
Yeah.
Yeah, it's not the right shape to fit into the enhancer. So, like allosteric effects are like the shape, based on the shape.
Uh-huh.
Now, if there's a steroid hormone involved, it binds to that green thing and it causes an allosteric change, which causes a change in its shape. So you see, the third thing, it's like shaped just to fit to the enhancer.
Okay.
Yeah. All right, you remember bioinformatics, where you just basically use computers and stuff, um, and all sorts of stuff like that. Okay, and then this is one of the techniques that are used called chromatin immunoprecipitation sequencing. So basically, you isolate chromatin from cell nuclei, you cross-link the DNA and the proteins with formaldehyde, and then you use sonication, which is basically using you know sound waves to fragment the DNA. So you break it up into parts. Then you allow an antibody that's specific to the protein of interest to bind to it. So some of the antibodies, let's say, it binds to that whatever bluish colored one. So yeah, we got those, and then we purify the complexes of with the antibodies, and we isolate, the protein of interest and DNA fragments. So we get the one that we want, and then we sequence that DNA.
Okay.
Okay. Chromatin conformation capture. Conformation is basically talking about the shape. Again, so chemicals are used to covalently cross-link proteins and DNA in close proximity within chromatin. So basically, we have that DNA loop because we use these proteins to cross-link, a piece of chromatin. And then we fragment the DNA, which means that we cut it essentially. Then we use DNA ligase to ligate, to tie together those ends. And then we get rid of the proteins, so we have just that circular bit of DNA that's purified. And then we can sequence that. And when we sequence it, we essentially identify the TADs, which are the TADA box, I think, activating domains, or transcription activating domains, I believe. So, so then that enables us to basically sequence it, and then figure out its position within the chromosomes. So we get that thing on the bottom.
All right.
Okay. Corrector, enhancer, promoter interactions require specific TAD boundaries. So, so mutations that disrupt TAD, which again I believe are transcription activating domains, can alter gene expression. One form of polydactyly, where you have an extra finger, or a couple of extra fingers, is due to deletion of a transcription activating domain boundary between those two genes. So basically a deletion there, right, which causes that orange thing to essentially turn on that IHH gene, which is normally turned off. Which causes, I suppose, another finger to be made.
All right, so that's it for that one. Okay. I'm sending you the some of the ones that we had done before towards the end, I guess, but we just didn't get through. It a lot, you and I maybe. I'm not quite sure. I think we did some of these, but I'm not sure. I remember only missing 2 or 3 with you.
Yeah. What are you're saying? You're sending it to my email?
Yeah, I'm gonna send you the other ones and stuff. There's 4 here, but I think we did 1 of them or 2 of them. But I'll go over them myself as well. But the test is cumulative and stuff. I feel like they're gonna go over Hardy-Weinberg and a lot of other stuff. So
okay.
From what I was looking at in this stuff, it's a lot of just the, genetics of regular stuff that we were doing, stuff that comes out much easier to us.
Yeah.
And stuff rather than the math portions, which get harder. So that's why I wasn't going crazy over these. I wanted to make sure I got an A in that other class
than my GPA.
Mm-hmm.
But the other stuff is easier to get, at least, because one of them is genetics of cancer, so that's more interesting and stuff, more engaging. Manipulating the genomes of eukaryotes, and they're not long; they're like 20 to 25 slides each, and then compared to like the other ones that we did, which were like 40 or
Okay.
45, so they're much smaller in
All right.
size. So I'm sending them over to you. Let me know when you get them.
Okay.
Right now. And I'll go over them myself as well. Some tomorrow, and then definitely Wednesday. And then on Wednesday, you'll be here at the house at 3 o'clock.
Yeah. When did you say your test was?
4 o'clock.
4 o'clock. Okay.
Yeah,
So
so 3, so we can
let's
just
see. These aren't
go over
crazy
anything
long.
that's.
Blah blah blah. No treads. Okay. Cumulative. Okay. 8. Short answer, but choose 6 to answer. Okay. So I see. Chapter 19, 14, 21, and 23. Did we not go over any of these?
I think we went over like one of them, and then the other ones I don't think we went over much. No. So I was just sending them to you so you kind of get familiar with it, but I'm pretty sure that we've explained it or like have gone over it in the past. You and I.
Okay, let's see.
Let's see.
Oh, this is gene regulation of
Chromosome warrior.
Okay, so I don't think we've done these then.
No, we haven't.
Let's see.
Crossing over.
We got 14, 21. If you could send them over with Google Drive, that would be easier.
Okay.
Because these,
do that.
well, I guess I could just upload them myself to Google Drive.
Okay.
But, okay, so this is all stuff that we haven't done before.
Okay.
Do you have any additional, like, quiz, like, practice, like, test kind of stuff?
I think we only got like 1 or 2 more, but I can send that too. Let's see.
Yeah.
They're very small. Let's see. What chapter?
Yeah, all these other ones are more in depth. It could take a while to go over them.
What happened?
These other PowerPoints you just sent me, they're more in depth. It might take a while to go over them.
Yeah. No, I know. I'm gonna be looking through them all pretty okay amount as well. So don't worry about that. Just send me these 4 for sure.
All right, I put them on whatever Google Drive. Wait, chapter 19, part 2. Is there a part 1 to that, or did we go?
We just went over part 1.
Got it.
So basically, part 2 just goes over the rest of it. It's like 12 slides or something like that.
Mm-hmm.
So it's not terrible. I'll be able to understand this. I read through some of them already anyway. I was making sure I'll be able to understand the pretty okay amount.
Mm-hmm.
So I'm just going to be focusing more on, like, I guess, the other stuff I wasn't super great about. I was just saying this to you because I know you know some of that stuff already.
Yeah, all
so
this stuff.
Okay.
So all this stuff in these PowerPoints you just sent me are, definitely stuff we have to go over.
Okay.
Let me see. Is there any time you can meet tomorrow, like online or something? So we can
can't.
go over these? Okay.
Not tomorrow, no. Wednesday would be like the only time.
Wednesday, could we meet like earlier? Just because, so these things are going to be on the test, and there's just like, we haven't gone over them at all.
Yeah. Right. So I guess
like in hindsight, we should have focused on these more than the labs, but it's okay.
Yeah.
So let's see, 2, 3.
Tell me when the test is from 4 to when?
It's from 4 to 5:40.
Oh, okay. So 4 to 5:40. So I usually come at from 4 to 7, I guess, for the 3 hours. 4 to 5:40 is like whatever, 2 hours. So I can come like, well, 3 to 5:40 is like 3 hours. 3. I don't know. Do you want to do like 2 or something?
2:30 to 5:40. That's like 3 hours and 10
Okay.
minutes. Does that work?
Yeah.
Okay, so 2:30 to 5:40. That works because we'll be able to go through that.
Um.
I'm just gonna go over past notes and stuff.
Yeah. Okay. So yeah, I'll
read
meet you at 2:30, and we'll go over these 4 PowerPoints. That's we're gonna have to go a little fast because there's 4 of them. So yeah, okay. I'll see you on Wednesday. Okay.
All right, Wednesday at 2:30. All right.
All right. Anything
See
else?
you then. No, everything else is good. You
All right.
doing all right?
Yep. Thank you.
All right.
All right. Take care.
All right then.
All right. Bye.
All right. Bye bye.